So we've talked a little bit about tamoxifen, and we've talked about CYP3A4 and CYP2D6. What I like about this next exercise is kind of the fact that it kind of puts it all together in terms of biotransformation, and there's immediate clinical relevancy. All right, so let's go through this exercise, and hopefully this will all kind of put everything into perspective. So tamoxifen is administered as a prodrug, okay, and it undergoes several metabolic steps. Two specific intermediate steps are shown here, but several CYP enzymes are responsible for these steps. And so the size of the arrow basically indicates the predominant pathway. So let's just follow this pathway along. So here's your prodrug tamoxifen, and with the help of CYP3A, undergoes chemical modifications into N-desmethyl tamoxifen, which is shown here. This is a intermediate. Okay. Now this intermediate metabolite undergoes another chemical reaction with the help of CYP2D6 into N-doxifen, okay, which is biologically active. Okay. So here's a potential major pharmacokinetic interaction looking at CYP3A4 inducers and inhibitors. Okay. So co-administered drugs can have a severe impact at this step right here. Okay. CYP2D6, okay, really has no drug-drug interactions in terms of induction, but has polymorphisms. Okay. So in the previous exercise here, okay, we were assuming that there was no other co-administered drugs, minus this red wing right here. All these other scenarios were basically looking at only tamoxifen, which would really indicate this pathway was unperturbed or unaltered. In reality, the breast cancer patient most likely is taking multiple drugs. And by chance, most of them are probably undergoing some type of biotransformation via CYP3A4. Okay, so there's one potential drug interaction. So assuming that these patients can make this particular intermediate without any problem, we should see, and the patient has normal CYP2D6 activity, we should see them making N-doxifen, which is biologically active. Okay, so if you have the patient on a CYP3A4 inhibitor, and you basically stop this cascade, there is this alternate pathway, which is here. So these other CYP enzymes, okay, CYP2C19, CYP2C9, and some of these others, can actually biotransform tamoxifen into 4-hydroxy-tamoxifen, okay, and then CYP3A4 can turn this intermediate into N-doxifen, okay. But you can see, I mean, in most of these situations, CYP3A4, and maybe CYP3A5, have major roles. So a CYP3A4 inhibitor is really going to alter the ability of any patient, okay, to make N-doxifen. All right, so that's just one way of kind of looking at this in a lot of detail to understand some of these major concepts. And this is pretty standard for most of the drugs, okay, where we've talked about maybe one or two different CYP enzyme systems being responsible for biotransforming one of these drugs into their components that are biologically active. All right, and so this is from a paper. So this was actually a real story. Here's a patient looking, three groups of patients being treated with tamoxifen, and what I did was I color coded it. So the black bars are the patients that have been identified after this study as being ultra-rapid metabolizers, the white bars being normal metabolizers, and this kind of bluish gray bar being the slow metabolizers. And so here's the drug concentration of tamoxifen. So you can see here that in terms of the administered dose, more or less all three patient groups received the same amount of tamoxifen. Okay. Serum concentration is about 160, 180 milligrams per ml. All right. And now if we look at the two metabolites, 4-hydroxytamoxifen and N-desmethyl tamoxifen, we can see that the the normal metabolizers and the ultra-rapid metabolizers made essentially the same amounts of this metabolite, but the slow metabolizers made I would say, a statistically significant reduction in that compound. Okay. Likewise, the ultra-rapid metabolizers didn't make very much of this particular N-desmethyltamoxifen, but these two patient groups made possibly the same amount. Okay, but these are the two intermediates, right? And these two intermediates do not have biological activity. The one that's key is this figure right here. Okay, so this is the amount of N-doxafen. So both the ultra-rapid metabolizers and the normal metabolizers, at the end of the day, made essentially the same amount of N-doxafen. Okay, this population, this patient population here in black, statistically had a much better response, probably because they made this amount of drug faster. Okay. This patient group, which is the normal patient group, had the same amount of endoxifen, but again, they made it probably a little bit slower. This group, look at this, had very little endoxifen being produced. Okay, and so in order to have a normal quote-unquote response rate, they needed to make this much. And so there's a big gap here in terms of the amount of endoxifen that they possibly could have made. Okay, and so these are the patients that, in spite of having that estrogen receptor breast cancer marker, they had a really bad response rate. Okay, and this is in spite of having good production of this intermediate. This intermediate is not biologically active. This one is. Okay, and so if you want to put the enzymes responsible for that, you can superimpose that, and of course, really it comes down to CYP2D6 activity, right? So in order for these patients to go from this metabolite, which they have plenty of, to this metabolite, which is biologically active, it's really dependent upon their activity of the CYP2D6 enzyme. And so these patients were identified as having very poor response because of slow metabolism of their enzyme system. So, from a clinical standpoint, why do we even bother administering tamoxifen? Just give the patients endoxifen and be done with it. Don't even rely on their metabolic activity. And so the reason we have to administer this drug as a prodrug, or the reason we have been, is because it's stable. Okay, and so what the medicinal chemists have done is that they've now made an endoxifen molecule that's more stable. Okay, they call it Z-endoxifen, and they've put it through phase 1 trials. Okay, to basically close this gap here, because we know that some patients just can't metabolize this drug. Okay, now we do this in the laboratory all the time. We'll physically make a drug biologically active because, especially if we're doing something with cell lines or in vitro assays, our cells don't have livers to fully conjugate all of these drugs, and so what we often do is we do the chemical conversion of these drugs in a test tube, and then quickly administer that chemical conversion in vitro. The problem is that these drugs are not stable for very long, and which is fine because these experiments sometimes don't go for very long either. So that's kind of the development of these drugs. You can look for more types of this studies coming out as the chemistry gets a little bit better. Okay, and so what does this mean? So this is kind of the birth of pharmacogenomics, okay, or personalized medicine. We've identified patients that just for a variety of reasons cannot take the standard protocol therapy, okay? And so what we're doing now is we're testing these patients before we give them the drug to kind of predict what the response rate is going to be. This concept has really been developed in oncology. St. Jude Children's Hospital has done some great work initially, and now lots of people are doing it. That's kind of the new thing now, where we can give another drug to kind of predict how efficient some of these patients are going to, metabolizing some of the chemotherapy. So a good example would be we can give codeine, and we can predict how efficiently they are going to biotransform Taxol. Taxol is a really expensive and quite nasty chemotherapy. Codeine is relatively cheap. Okay, so if we can identify the ultra slow metabolizers of codeine, we can also predict some of the toxicities that might develop in pediatrics. We also look at some polymorphisms for warfarin. And what does this mean? Basically, if patients are resistant or have resistance to some of these drugs, they're going to spend a lot of money, and it's going to delay the appropriate medical care. And it really is kind of the wave of the future. And you can probably understand that. So these polymorphisms are going to be more able to be tested in the next couple of years. Now, there's companies out there that are offering to look for some of these polymorphisms, especially with CYP2D6, which we had just talked about, as well as CYP2C19. And it's these polymorphisms that actually kind of account for the narrow therapeutic index that's experienced by at least some of the population that are being treated. Okay. Now they've got some of these cytochrome P450s on DNA chips, and so what we can do is we can actually put the drugs on there in an in vitro assay and see how quickly they are actually biotransformed into their specific metabolites. We can pre-test people, and we can predict adverse drug reactions. And so this is kind of the wave of the future, and you'll see a lot more of this in your clinical practice in the upcoming years. Okay. So other things that can affect the efficacy of the drug include underlying diseases. This particular block is all about food to fuel, and so underlying metabolic changes, cirrhosis, alcoholism, hepatitis, those things can severely restrict a patient's response to a given drug because their liver physiology is abnormal. And of course, we said a lot of these cytochrome P450 enzymes reside in the liver. Okay, and so the other thing I wanted to point out is this really nice summary table shown here, looking at all the different cytochrome P450s and the drugs that are substrates to them, drugs that are inhibitors, and drugs that are inducers. Okay, and so we'll keep coming back to this. No, you don't need to memorize this, but you will need to learn it, and we'll learn it throughout the curriculum. And I think by the end of the year and by the end of your clinical rotations, after talking about this so many times, you'll know a lot of these drug interactions. All right, thank you.